Thank you for the very kind introduction. So today I'm going to be talking about one strand of my work, which is on using genomic data in social science, or moving toward the world where genomic data can be used in social science. So the work I'm going to talk about is the product of many people's efforts. It's highly collaborative, interdisciplinary work. Dalton should be on this list. There's just not enough space on one slide to describe the dozens of people who contribute to the work I'm going to be talking about. And this, from economics, sociology, psychology, statistics, genomics, it requires a lot of different perspectives. Now, what motivates me in wanting to combine social science and genomics is the prospect of allowing us to do better social science. And I think there's a number of kinds of ways that genomic data could help with social science. I've listed a few of them on some of what I think are some of the most important on the slide. But so for example, studying genomic data can help us learn about the mechanisms underlying behavior. In one recent paper that my collaborators and I did, which was focused on subjective well-being and depression, when we looked at the results that came out of genetic analysis, what we found is that a lot of the genes were involved in the glutamate receptors, which is actually a bit of a surprise because a lot of the literature had been more focused on monoamine neurotransmitters. So that's the kind of thing that you can learn from genomic data that could then guide work on biological mechanisms. A second kind of application for genomic data, or use of genomic data, is to help us do empirical work that may itself have nothing to do with genetics. For example, if you have a genetic variable that is sufficiently predictive, and I'm going to talk later about polygenic scores, which are indexes of genetic variants that combined can have substantial predictive power, like a polygenic score for educational attainment that explains 10% of the variance in people's education. Then you could use it as a control variable in any study that is just trying to estimate a treatment effect on educational or achievement outcomes. So let's say, for example, you do a randomized experiment where you're interested in the effect of providing free preschool to kids. That kind of experiment is very expensive to do. Sample size and power is really at a premium. Having a powerful control variable that you can collect inexpensively, like get a saliva sample and genotype the people and then construct a polygenic score and control for it, can dramatically improve your power. So even if you're not interested in the effects of genes, just having a good control variable can be very useful. Of course, you might also be interested in a non-experimental setting in controlling for genetic factors in order to unconfound genetic and environmental factors, and that would be another use of genetic data as a control. In some special circumstances, you could think of using genes as instrumental variables. I know, Dalton has thought a lot about when this may or may not be appropriate, but I'll give one example where arguably the assumptions required might be met. Would be a case where you have a gene that's a nicotinic acetylcholine receptor gene that's involved in the metabolism of nicotine. Those genes are pretty well understood biologically, and we believe that they affect how quickly the body metabolizes nicotine. So you could use such a genetic variant as an instrument for smoking if you then want to study the causal effect of smoking on something else. And you can may be able to argue in that kind of situation that the gene is unlikely to directly have some other direct effect on the outcome. And finally, we're often very interested in studying gene-environment interactions or relatedly targeting interventions. And having direct measure, you could do that to some extent with twin and adoption studies, but having direct genomic data allows much more, a wider range of study designs, often with, allows you to ask questions you couldn't otherwise ask. We could think about using the genomic data to target interventions, like if we knew who was going to be at risk of declining cognitive health at older ages, help those people, give those people advice to prepare early for what's coming. So, so these are motivating kind of applications of the data. I'm actually not going to be talking much at all about these. I'll come back to them a little bit at the end. My research has been focused on the logically prior question of which genetic variants are related to behavioral outcomes. How can we construct the predictive variables that we could use as control variables or use to do gene-environment interaction studies? And this is ultimately what motivates me, and I'm doing my best to encourage good work using the results that we're producing. But we've been at a stage in the last few years where we haven't yet had the tools to be able to do these applications. And so we're trying to create those tools to allow this kind of line of research to emerge. So I want to start the talk with a very quick genetics primer, and then I'll talk about the kind of the story of my own research in this area, and then end with some recent, not yet published results on the genetics of educational attainment. So I should apologize that this primer is going to be way too elementary for those of you who actually know something about genetics. So I'm going to oversimplify the world, and apologies in advance for that. But it should be just enough background to be able to follow what I'm going to talk about later. Okay, so DNA is a sequence of 3 billion pairs of nucleotide molecules that are spread in humans across 23 chromosomes. Most of that DNA is actually not genes, but there are subsequences of these nucleotide molecules which are genes. There's about 20 to 25,000 of them. And it's those genes that provide the instructions for building proteins that then go into the body and do things. The rest of the DNA may be involved in regulation. Some of it is involved in regulation. Some of it's just not known yet what it's involved in. At most of these locations in the genome, there is no difference across people. It's just the DNA is providing the instructions for how you make a human being. What we're going to be interested in is the small fraction of locations in the genome where there are differences. And those are called polymorphisms. And I'm going to talk in this talk as if there's just one kind of polymorphism, which is a single nucleotide polymorphism. And that's a location in the genome where the nucleotide molecule, the single nucleotide molecule differs between people. There are other kinds of genetic variation. Most of the important ways, they're very similar to single nucleotide polymorphisms or SNPs, so I'm just going to ignore them for now. Now, at virtually all SNP locations, there are only 2 possible nucleotide molecules. So these nucleotide molecules are the A, C, G, T letters. So at a particular location, there's only 2 of them, say like a C or a T. And one of those is going to be more common in the population. We call that the major allele. And whichever one is less common, we call the minor allele. And each person gets one allele from each parent. It could be the major allele from both parents, or the major from one and the minor from the other. And the function of the SNP doesn't depend on who you get the allele from in most cases. And so we can summarize your genotype at a given location by the number of minor alleles, 0, 1, or 2. So what this means is that when we talk about genomic data, it's going to be a long vector of SNP locations. Those are the rows. And each entry is going to be a 0, 1, or 2. Now, there's different kinds of genomic data. The kind that you probably hear about most commonly is what's called sequencing data. And sequencing is what happened when the Human Genome Project was completed around 2001. And that is when you measure pretty much every nucleotide in the genome. It's the entire DNA sequence. This graph shows the cost. The y-axis here is on a log scale. So part of the point of this figure is that the costs have been falling extremely rapidly. So in 2001, when the Human Genome Project was finished, it cost $100 million to sequence a human. Last year, it cost $1,000. And this rate of decline is faster than Moore's law. It's just extremely amazing. It's amazing the rate of progress in the technology. Now, the kind of data that I'm going to be talking about is not that. It's genotyping data. So genotyping data is where you focus just on the SNPs, so just in locations of the genome that differ across people, and not even all of those. So typically you measure 2.5 million SNPs, and there's something like 50 or 60 million in the genome. But those 2.5 million are chosen in a way that they capture almost all of the variation across people. Because the way that the DNA gets inherited from parent to child is it gets inherited in chunks, which means that there's correlation in the genome. It's called linkage disequilibrium. And so by measuring these 2.5 million SNPs, you can infer what the other ones are to a pretty high degree of accuracy. And so you know pretty much everything you need to know about the differences across people. So the cost of genotyping has also been falling very rapidly. As of last year, recently, it costs less than $50 a person to get genotyping data. And the cost has been falling by roughly half every year. So it's, in a few years, it's going to be the main cost of collecting genotyping data is going to be the cost of paying the human subjects to spit in the tube, and maybe the cost of the postage to send the sample to the lab and get it back. The actual genotyping is going to be small relative to that. Okay. So traditionally, the way that we tried to study which particular genes were related to behavioral traits or disease traits was through what's called a candidate gene study. So a candidate gene study is where you specify some hypothesis, sometimes just one, about just one SNP or a small set, based on what you think the biological function is, and then you test whether in fact that SNP is associated with the outcome of interest. Sometimes these are interactions. Sometimes you also include interaction terms if you have a hypothesis about a gene-environment interaction. And you set the significance threshold typically at 0.05, the usual level, or sometimes you adjust for multiple testing if you test more than one SNP. And this characterized virtually all of the work in medical genetics up until about 2005, and still characterizes most of the work in social science genetics. Before 2005, it was just too expensive to collect genome-wide data. So you pretty much had to focus on just a small set of genetic variants in order to be able to afford to do the work. Now, this is, of course, a totally reasonable way to proceed. It's the usual way that we learn about doing science. You make a hypothesis, and then you test it. And there are classic examples where it's worked very well. For example, the discovery of the association between ApoE and Alzheimer's. ApoE is the strongest genetic predictor of Alzheimer's. It was discovered because it was known what this ApoE gene did. It coded for a particular kind of beta amyloid protein that was known to be present in the plaques that build up in the brains of Alzheimer's disease patients. And so it was a reasonable hypothesis to test that variation in that gene might be related to Alzheimer's. And in the early 90s, that was tested and confirmed. there are other examples of that kind of success, and that helped fuel interest in the candidate gene approach in social science. But in the later applications of candidate gene approaches in medical genetics, and in, as well as in social science, a lot of the findings haven't replicated very well, or at least the replication has been inconsistent. When I got started in this area, so I was interested basically from the beginning of graduate school, which for me was in 2001. At that time, there was no data available at all, and the norm at the time in the community was that researchers collected their own data and were very protective of it and didn't collaborate with anybody. So it was basically impossible to do anything when I first got interested. Around 2004, I was working with my advisors in graduate school, David Leibson and Ed Glazer, and we got access to data from the Icelandic Heart Association. And at that time, candidate gene studies were the way that you did this kind of work, and so we did one ourselves. We came up with a list of dopamine and serotonin and other genes that were known to be involved in brain function. And we measured them on a sample of Icelanders, and we had a survey that I took some trips to Iceland and put together all the variables that were of potential interest to social scientists, and then we did the usual thing at the time, which was lots of multiple hypothesis testing. We looked for whatever kind of genes related to whichever kinds of outcomes, and we found something, and we were very excited. It was an association with educational attainment. And we came back to our medical collaborators. Now this was 2007, 2008, and by then things had changed in the medical world, and there was a lot of skepticism about candidate gene studies. And they said, you have to replicate this before we can try to publish it somewhere. So we went to 3 other data sets, including one other data set from the same sample in Iceland, and got absolutely 0 effect, so no replication at all. So this was my first introduction to this world of genetics. It was pretty depressing. My collaborators and I spent a long time trying to understand what was going wrong, and my colleague Chris Chabris, who's a psychologist, led a study that we did on looking at systematically at candidate genes that have been found to be associated with IQ and testing whether they replicated, and when we tested them in much larger samples. We put together a sample of 10,000 people by meta-analyzing data across 3 data sets, and 10,000 people was much larger than most of these original studies, which were based on a few hundred people. And we looked at 12 different genetic variants that have been found in the literature, and none of them replicated. And that paper was published in 2012. At that time, social scientists like us were starting to realize that there were some problems, potential problems with this candidate gene approach. It was also, I don't think it was not broadly recognized. I think it still isn't, completely, fully broadly recognized. But there were corners of the social science genetics world that were starting to understand this. One of the field journals, lead field journals, Behavior Genetics, came out with a new editorial policy in that year, 2012, that imposed much stricter standards on candidate gene studies. It said you had to accompany them, for example, with a replication. You had to do power calculations and show that the study design was adequate. And this new editorial policy was accompanied by a very harshly worded introduction, so I'll just read the first few sentences. The literature on candidate gene associations is full of reports that have not stood up to rigorous replication. This is the case both for straightforward main effects and for candidate gene by environment interactions. As a result, the psychiatric and behavior genetics literature has become confusing, and it now seems likely that many of the published findings of the last decade are wrong or misleading and have not contributed to real advances in knowledge. And then it goes on to talk about why that was true. This editorial policy was subsequently adopted by some other journals in psychology, including Psych Science, which is a top journal in psychology, and the journal Emotion, and there may be some others. Okay, so why did the traditional candidate gene approach have problems? Well, I think there's basically 3 issues. One is multiple hypothesis testing, which I think we're coming to understand is a much broader problem in social science and in science, but it was also a problem in the candidate gene literature. There were typically many genetic variants that were measured, and often several phenotypes that could be studied in different ways you could specify the regression that you run. And they usually weren't all reported in the paper, or that you would write a paper about the relationship that actually worked, and that wasn't adjusted for in interpreting the results. Second issue is population stratification. So this is the single biggest confound that comes up in genetic association research. The issue here is that your genotype may be correlated with your ancestry. It will be correlated often with your ancestry, which is in turn correlated with environmental factors that actually matter for the outcome, and it's not the genetic variant. So a classic example of this is the chopsticks gene. Let's say you were doing a study to try to find which genes explain the use of chopsticks. So obviously that's not genetic at all, it's totally cultural. But there are plenty of genes that vary, the genotypes vary in frequency across Asian and non-Asian populations. And so any such gene like that is going to show up as spuriously associated with chopsticks use. And that's very hard to deal with in the traditional candidate gene study, where you've typically just measured a few genetic variants. Controlling for self-reported ancestry is not sufficient because there's variation at the sub-ethnic level. Or restricting to whites. Whites is too broad of a category. There's lots of variation among ethnic groups among whites. The third problem, which I want to spend a little bit more time on, is low power. The issue here is that virtually all, up to I think it's essentially true that all of the candidate gene studies had samples in the range of 50 to 3,000 in social science. Many of them were laboratory studies with a few hundred subjects. And so doing a study with that sample size involves an implicit assumption about the magnitude of the effect. So something like an R squared of at least 2% to be reasonably well powered. But there's plenty of reason from the physical and medical literature to be skeptical that those are really the effect sizes for particular genetic variants. So for example, the association between smoking and the SNP and the acetylcholine nicotinic receptor that I mentioned earlier, that's the strongest genetic association with smoking, that has about a 0.5% R squared. The association between BMI and FTO, which is the strongest genetic association with BMI, is about a third of a percentage point R squared. And for height, where there's now over 700 SNPs that are known to be associated with height, the largest of them has an association with a strength of R squared of 0.4%. Now, back in 2012, not all of this was known, but there were numbers bigger than this that were credible numbers from the medical and medical genetics literature. So even back then, if we think that the associations between genes and behaviors are, if anything, probably smaller than these more direct biological effects, we should have been skeptical about findings or study designs that were only powered to detect effects larger than 2%. Now, over the last few years, we have credible evidence from large-scale studies of behavioral phenotypes, and they confirm, in fact, that the effect sizes are actually much smaller for behavioral traits. So, for example, for educational attainment, the largest effects are about 0.02 of a percentage point R squared. So, an order of magnitude smaller than for the medical traits. For depressive symptoms or personality trait neuroticism, it's about 0.04 of a percentage point R squared for the largest association. Subjective well-being is also about 0.02 of a percentage point. These are, again, for individual SNP associations. So let's think about what that implies for the credibility of the traditional candidate gene studies. Suppose that we have a significance threshold of 5%, standard significance threshold. Suppose the strength of the association is 2/100th of a percentage point r squared. So what we're getting for educational attainment for the largest association. And let's say you do a big traditional counting gene study with a sample size of 3,000 people. So then your power is about 12%. And what kind of sample size would you need for 80% power? Well, turns out you'd need almost 40,000 people. So an order of magnitude bigger than the biggest traditional candidate gene studies that were being done. Now, we can also ask another question, which is a Bayesian question, about the credibility of a finding that's significant at the 5% level. So the question is, suppose somebody comes to you, maybe you're reviewing a paper, or or you find the result yourself and you're not sure whether to believe it. It's significant at the 5% level, and you want to ask, what's the likelihood this is a true positive as opposed to a result by chance? So we can do this calculation for different prior probabilities that there truly is an association. So I think that the range of 0.1% to 1% is a plausible range if you're just picking a SNP at random in the genome for a trait like educational attainment. 10% I would consider to be a pretty optimistic prior for a complex trait where you don't have detailed knowledge of the biological pathway. We can do the calculation for 3 different samples, for whatever sample size. I'll show you for 3 sample sizes. 100 is like a lab experiment. 10,000 would be for, say, a social science survey. And 100,000 would be like a kind of consortium meta-analysis like I'm going to be talking about soon. So looking first at a sample size of 100, the power here is just a little bit more than 5%. And in this case, even if you start with an optimistic prior of 10%, and you find a significant result at the 5% level, should barely move your beliefs at all that there's really an effect. So why is that? Well, the intuition is that the significance threshold of 5% means that the chance you would have gotten a result like you did if there were no effect is 5%. Having power of 5% plus epsilon means that if there were an effect, the chance you'd find a result like you did is just a little bit more than 5%. So having found that effect, you barely update at all, because it's about equally likely whether the null is true or the alternative is true. Now, interestingly, even if you have a sample size of 100,000, where your power is 99%, and even if you have an optimistic prior of 10%, there's still a significant chance of a false positive. You should only be about 70% sure that the result is true when it's significant at the 5% level. And the reason for this is that you can only get, if your significance threshold is only 5%, you can only get so much evidence from finding a significant result. Because the chance you find the result if there really is an effect is almost basically 100%, but the chance you'd find it if it's not real is about 5%. So finding an effect, finding a significant result is like 20 to 1 evidence that there really is an effect. But if your prior is as low as 10%, that doesn't move you all the way up to like 95%. So in a world where priors are small, you actually need more significant thresholds than more stringent significance thresholds than 5% in order to get convincing enough evidence that there really is an association. So one of the lessons from genomics is, we're in this world of small priors. We need to think about adopting different standards for significance. Okay, so in 2005, I think it's fair to say this was pretty well understood in the medical world, and there was a shift in the kind of study that was done from a candidate gene study to a genome-wide association study. So what is a genome-wide association study, or GWAS? It's where you get this genome-wide data that I talked about before, which these days is about 2.5 million SNPs, and theoretically, you just test each of them for association with the outcome. So you don't pretend that you have any kind of adequate biological knowledge to be able to focus on a small number. You say, let's see what the data tells us. And then you set the significance threshold at the level of, instead of 0.05, 5 times 10 to the negative 8th. That's called the genome-wide significance threshold. And there's a couple of ways to think about it. One way to think about it is it's a Bonferroni correction that's appropriate for European ancestry populations where there's about 1 million units of independent variation in the genome. So I mentioned before that there's about 60 million SNPs, but there's correlation among the SNPs, so you can calculate what the effective total amount of variation is. It's about 1 million. So this is the, roughly the Bonferroni correction that you would need. There's also a Bayesian argument along the lines of what I was just talking about, that we want to, because our priors are really low, we want to have a really stringent significance threshold so that significance provides enough evidence that there really is an effect to overcome our low priors. Now, if you have that kind of significance threshold, of course, we had a power problem before at a significance threshold of 0.05. Now we really have a power problem. We're going to need much bigger samples. And so along with the shift to genome-wide association studies, there was a change in the culture of research in medical genomics, where the studies went from being, being protective of their own data and not wanting to collaborate, to realizing that to publish, they had to meta-analyze, pool their resources and meta-analyze their results across many studies. And so, so there emerged consortia that allow you to get sample sizes in the tens of thousands or potentially hundreds of thousands of people. And then there was also, because you had to coordinate across so many different samples, you had to write your analysis plan in advance to make sure that every study did exactly the same thing. And so it became very easy to have a pre-analysis plan that then was straightforward to pre-register. And so that just became the normal way that people did work, because they had to coordinate what everyone was going to do. Okay, so how does this help with the problems I mentioned before? Well, with multiple hypothesis testing, we're now looking at all the measured SNPs, so it makes transparent the multiple hypothesis testing problem, and it's addressed with the genome-wide significance threshold. For population stratification, for population stratification, I mentioned that controlling for self-reported ethnicity is not good enough, but what you can do when you have genome-wide data is you can actually estimate ancestry from the data itself. You can look at principal components, which is a measure of the kind of joint dimensions of variation along which people differ in their allele frequencies across many genes, and that tracks ancestry, it turns out, very well. In fact, that's how companies like 23andMe and tell you your ancestry based on your genomic data, is they're estimating principal components. And so you can do that and then control for them. And so that actually provides you pretty high resolution control for ancestry. It's not a perfect solution to the problem of population stratification, but there are other things you can do too that you could, for example, look at sibling samples and put in sibling fixed effects. So effectively look at whether differences in genotype across siblings predict differences in their outcome. Then you've perfectly controlled for ancestry because you're looking just within a family. That's not possible to do in sufficiently large samples to discover gene associations, because there just aren't big enough sibling samples with genotype data. But you can do it as a robustness analysis. Once you've found the association, then you can look at a few of these variants in sibling samples. Okay, and then to deal with the low power, we have this consortium structure. So let me come back now to the kind of calculation we did before, but now looking at genome-wide significance. So the only change here with this Bayesian calculation is that we're, instead of 0.05, we're doing 5 times 10 to the negative 8th for the significance threshold. And you can see that a sample size of 100 is basically hopeless. Your power is basically 0 for detecting a true association. Even with a sample size of 100,000 with such a stringent significance threshold and an effect size of 0.02%, your power is only 16%, which means that you're only going to discover 1 out of every 8 SNPs that are truly associated with educational attainment with this kind of study design. But even with a very low prior, if you find something, it's virtually certain to be a true positive. Okay. So now I want to make some of that more concrete and talk about the example of educational attainment, which is one of the phenotypes we've been working on in the last few years, and I think this, the behavioral phenotype where we've made the most progress to date. So I told you about this sad story of my collaborators and me and our failed replications, and that was basically in 2011. I actually gave versions of this talk back then, and they were super depressing, and they ended with failed replications. I'm happy to be able to tell you that this current version is going to have a more optimistic ending. But in 2011, David Cesarini, who was the speaker here last week, Phil Kohlinger, and I founded the Social Science Genetic Association Consortium. And basically what we wanted to do was imitate what was working in medical genomics. So they had made this shift to genome-wide association studies through consortia, and we thought this is our best chance for making progress in social science as well. The first question that we faced was, what outcome should we study? We did a lot of power calculations, and there were there were arguments that maybe we should be focused on something like cognitive ability, which is measured by hour-long tests and has low measurement error and can be measured pretty reliably, or personality traits. Or should we, but the problem with those kinds of outcomes is that they're not measured in very many samples, partly because they're so intensive to measure, and the medical geneticists weren't interested in those outcomes, so they weren't measuring them. So that might have been a sample of 10,000 people. Or we could study something like educational attainment, which had the key advantage that it was measured in all of these medical data sets, because people studying cardiovascular disease and cancer and everything, they were measuring education as a background variable, so they had the genome-wide data. The problem there, though, is obviously educational attainment is extremely far from what the effects of genes do. It's highly socially influenced, and there's different institutions across different countries, and people told us at the time that it was crazy for us to study education, that we were throwing our careers away by making that choice. But we did the power calculations, and we came to the conclusion that if we could get 100,000 people with educational attainment, we were actually going to have a better chance of finding something than studying even 25,000 people with a kind of better measured variable. So we decided to pursue educational attainment. The first paper, which we now look back on and call it EA1, was, and we ended up recruiting from among medical genetics data sets, 41 data sets with a total sample size of 100,000, which was, really just a totally different order of magnitude than any of the work in social science genetics up to that time, and similar to the kinds of sample sizes that were available now for the largest, at the time, for the largest medical studies. We looked at 2 outcome variables, a measure of years of schooling and a binary variable for completing college. We did this kind of study the way these consortium studies are generally done, which is we had an analysis plan to make sure that every genome-wide association study in each data set was done the same way with the same kind of controls. Which, by the way, they're because you're trying to make it doable for all of these data sets, it means you don't have that many controls. It's not very good social science in the sense of modeling the environmental heterogeneity or anything. It just kind of the dirtiest thing you can do that's halfway reasonable, like control for sex and age, and make sure you map educational systems in different countries onto a common international standard. So each of these studies did their own analysis, and then they uploaded their GWAS results to a secured central server. And typically, then they make tons of mistakes, and we don't have access to the individual level data because of privacy and IRB concerns. So we just have to tell them what analysis to do, and they can try to diagnose where the errors are. And we're just looking at the summary statistics and trying to figure all this out. So this takes months of back and forth. Once we are sure we have clean results, then we meta-analyze the GWAS results to obtain the overall regression coefficients and standard errors. And what we found in the end was one genome-wide significant association with the years of education measure and two with the college measure. And then following what had become standard in the medical genetics literature, we had a replication phase of the project where we recruited 12 more data sets that, there's more and more of these data sets that are getting genome-wide data then and now because it's getting so inexpensive and so there's more of this data becoming available, so we could recruit new data sets that weren't available for the discovery part of the project. And this sample size was 25,000 people, and all three of the associations replicated at the Bonferroni-corrected 5% level. So this paper was published in 2013 in Science. This is a Manhattan plot. So this is the typical way you'll see the results from genome-wide association studies illustrated. What is happening on the x-axis is it's the whole genome laid out across all the chromosomes, and each of the points is a SNP. And the reason it looks like just a solid mass is because there's so many of them, so they're all lying on top of each other. The y-axis is significance. It's the p-value on a negative log 10 scale, so that higher numbers mean more significant. And this bar here is the black bar is the genome-wide significance threshold. So what you see in this kind of figure is there's lots of these towers that show up, and this is called a Manhattan plot because of the towers look like the skyline of Manhattan. And the reason why there are these towers is because of the correlation in the genome. So when one of the SNPs is highly associated with a trait, it's highly correlated with nearby SNPs, and so they also tend to be highly correlated. And what you see here is this is the results for college. You see these two towers here that peak above the genome-wide significance line. Those are the two hits for college. And one thing you can understand easily looking at a figure like this is that what you find in a genome-wide association study is not a particular SNP or a particular gene that's associated with the trait. What you're really finding is a region. We're saying there's something in this area here where all of these SNPs are correlated with that's associated with the outcome. And you have to do follow-up analysis and maybe study the biology and try to figure out where is actually the causal SNP or the gene that matters. Yeah?
Either of these two was the one that was correlated with years of schooling? So it sounded like
there was a third one that was correlated with years of schooling. I think it's this one here that doesn't reach significance in college. Okay, so that was in 2013. There's lots of other analysis in the paper. I'm just going to, for time, I'm going to skip to the next part of the story, which is we call EA2, which we were able to do a few years later. It's published last year. We were able to increase the sample size because, again, there's more and more data sets with genome-wide data. But now we have 63 data sets with about 300,000 people. We decided now to just focus on years of schooling and forget about the binary college variable. And now we find 74 SNPs, independent SNPs that are associated with education. And while this paper, EA2, was under review, a big new data set became available called the UK Biobank. It's now 500,000 people, but the first initial release was 110,000 people, or 150,000, 110,000 of which had data on educational attainment. And we were then were able to use that data in our revision of the paper as a replication sample. And the replication was extremely strong. So 72 out of the 74 hits had the same sign in the replication. 52 of them were significant at the 5% level. 7 of them reached genome-wide significance. And what we can do is compare this replication record to what you would expect given sampling variation and this kind of size of the replication sample under the hypothesis that all of the effects were true hits in the original discovery. And it turns out this replication record is, if anything, a little bit better than you would expect if all of the original hits were true. Yeah.
What's the reason for doing this as a replication rather than integrating the data with the large data set?
We also did that. So with that, you get 162 hits. But this was a way to test kind of robustness.
Exactly.
Yeah. So this is, okay. Actually, I thought, sorry, these slides are a little out of order. This actually shows the replication. This shows for each, the 74 SNPs are on the x-axis here. The y-axis is the effect. Here I'm showing it in years of schooling. The black points, 95% confidence intervals, are the effect sizes in the discovery sample of 300,000, and the blue ones are from the 110,000 replication sample. So you can see visually the effect sizes are generally quite similar across the two. Okay, so that paper was published last year in Nature. We've been, partly because that paper took a very long time to get published, and partly because just the tremendous growth in the availability of data, by now we've actually been able to further expand the sample size dramatically. So we're going to tell you now about work that's still in progress. We now have a sample of 70 data sets with 1.1 million people in educational attainment. And now we're finding 1,271 independent SNPs. So here's the Manhattan plot. It's actually totally uninformative now with this kind of scale of the data because basically it looks like everything is significant, which it isn't. It's just that you can't tell the difference between the places that are significant and the places that aren't. Finding those SNPs is very useful for some of the kinds of analysis we want to do with GWAS results. So for example, the biology, learning about the biology, having lots of hits gives us a lot of information to study which genes might be related to the outcome. And we do that in these papers and find some interesting things. For example, as you'd expect, the genes related to educational attainment are very highly expressed in the brain. But it turns out that glial cells don't seem to be involved. That basically, the genes that have differential expression in glial cells don't show up as particularly strongly related to educational attainment. So it doesn't seem to be about that part of the brain. So I'm not going to talk about the biology here, but I want to talk about now, just tell you a little bit about this other thing that comes out of these GWAS studies that I think is the most useful thing for social science, which is the polygenic scores. So these are not the individual SNPs, but the indexes of many SNPs that when you combine their predictive power can add up to a lot, and you can use for control variables or to do well-powered gene-environment interaction studies, for example. So what I'm showing you here, and basically the way you construct these polygenic scores loosely is you take the regression coefficients that come out of the GWAS, and you adjust for the correlation across the fact that the SNPs are correlated with each other. And then you basically created a weighted sum of the genotypes, where you weight by the effect size of the SNP. And so you get a maximally predictive linear index of the genome-wide data for predicting the outcome. So what I'm showing you here is how much predictive power you can get from these polygenic scores, and how that's changed as our sample sizes have gotten bigger for the GWAS studies. So this is our EA1 polygenic score, and it's showing you this for two different, so the x-axis here is the sample size of the study, the y-axis is the predictive power of the polygenic score in terms of R squared. I'm showing you results for two different validation samples, the AdHealth data set, which is a sample of young people that's nationally representative, and the HRS is a sample of people over the age of 50 that's nationally representative. And so they're not included in the GWAS discovery sample, so this is an out-of-sample assessment of their predictive power. From EA1, we were getting 3 to 4% predictive power for the polygenic score. For the 300,000 from EA2, we were getting about 6 or 7%. When we combine the data, as you were suggesting, we have about 400,000 people, then it goes up to about 7 to 8%. What we're now getting in EA3 is between 10 and 12% R squared. So we're explaining, I just want to repeat that because it's actually pretty remarkable. 10 to 12% of the variance across people in their educational attainment is being explained by the genome-wide data alone. So that's huge compared to any kind of effect sizes you typically get in in studies of education. Here's another way of looking at that kind of level of predictive power. Here, what I'm doing is cutting the sample, cutting the polygenic score into quintiles. So this is the lowest fifth of the polygenic score distribution. This is the highest fifth. And the y-axis here is the frequency of completing college. So depending on the data set, it's about 10% of the people complete college who are in the lowest polygenic score quintile. The highest polygenic score quintile, it's between 40 and 55%. So, huge, huge amount of predictive power for college completion. This, what I'm showing here on the top, is comparing the predictive power of the polygenic score against other common predictors of education. So you can still predict a higher amount of variance if you use cognitive ability or measures of parents' education. They predict a little bit better than our best current polygenic score. But we're predicting more than you can predict with income. Okay. So let me, there's a lot of other things in the paper which I won't have time to talk about. Let me just mention a couple of things. One is, when we started this work, one of the big concerns that we heard was that there's a lot of heterogeneity in the environments, and we should expect the effects of genes are going to vary a lot depending on which country we're talking about, and even within a country. And that's a totally valid, reasonable concern, something we needed to try to account for in our power calculations. And now we have evidence for it. We can quantify the extent to which that's true. We can look at things like how much heterogeneity is there in the genetic effects. So we can look at, this is the average genetic correlation, which is, roughly speaking, it's asking for a given SNP, how correlated is its effect in one data set with its effect in another data set? And the answer is, significantly different from one. It's 0.7. So there's substantial heterogeneity. And the thing, one of the big takeaway messages from this talk is that with the levels of predictive power we're now getting from the polygenic score, the kind of promises that I started out with, the reasons why we might be excited about genomic data in the social sciences, they're now real. Like we can really start doing them. So for example, with a predictive power of 10%, if you wanted to study the effect of the polygenic score for educational attainment, you'd have 80% power to detect its effect at the 5% level in a sample of only 75 people. So we can now go back to the lab. We can do laboratory studies like we were trying to do with candidate gene studies, but do them with polygenic scores and be well powered. We can also start using these polygenic scores as control variables. So if you're increasing your variance explained in education from 10% to 20% by including the polygenic score in your randomized experiment, you're reducing the required sample size for any given level of power by 11%. And that can be real cost savings with an expensive kind of intervention. Now I've been talking about education, but with the SSGAC, we've been studying a lot of other phenotypes as well. So fertility, subjective well-being, risk tolerance, dietary intake. And so these and other phenotypes are ones where in the near future we'll also have highly predictive polygenic scores. And so let me just end by commenting that I think we're starting to see a real change in social science genetics. So we know of at least 74 ongoing projects that are using the educational attainment polygenic score. We know that because on our website where we make the results publicly available, we ask people to describe if they're going to use the data for something so we can try to keep track. There's some really interesting papers that have been coming out. For example, there's a paper that Dan Belsky and are you a co-author of Dalton? No. Okay. But there's a number of it's a really good paper that came out in Psych Science last year, looking at the educational polygenic score and trying to map out how what it predicts over the life course to understand the mechanisms by which the genes seem to be mattering. So finding things like it's related to early speech acquisition and its effects seem to be largely operating through cognitive ability, but also through some personality traits like openness to experience. There's also now work using the polygenic scores to do well-powered, well-identified gene-environment interaction studies. So for example, my colleagues, some of my colleagues have a paper where they're looking at the change in the compulsory schooling law in the UK, where people were required to go to an additional year of schooling, 8th grade and 9th grade, and looking at the effects, using that to look at the causal effect of education on health outcomes like BMI. And prior work has found that the average effects on health outcomes are really small and not statistically distinguishable from zero. But what they're finding is when they use a polygenic score for BMI to look at heterogeneity in the effect, there's really big differences, that there's basically very big effects in people who are at the high end of the polygenic score distribution. So people who are genetically predicted to have high BMI are really affected a lot by that additional year of education. It reduces their BMI by a lot. And it's the people at the low end who weren't predicted to have high BMI anyway who were barely affected at all by the additional year of schooling. And I think this is, the amount of work in this area is just going to continue to mushroom in the coming years because the genome-wide data is becoming so inexpensive to collect, available in so many more data sets, easy to collect it yourself in a lab experiment if you want. And our genomic knowledge is correspondingly growing very quickly. And so we're going to be able, in the very near future, to construct predictive polygenic scores across a very wide range of traits that we'd be interested in. Let me stop there.